Hi, my name is Demetra Mangum. I'm a senior biology pre-physical therapy major at Alcorn State University, and today I will present my senior project, the annotation of genes of the Halothiobacillus Neapolitanus C2. It is a proteobacteria that is also gram-negative. The Halothiobacillus is also an anaerobic bacteria, which means that oxygen is not required for growth. It's a sulfur oxidizing chemoautotroph, which means that it's rich in sulfur, and the bacteria depends on carbon dioxide for energy, which is part of a natural gas that helps produce energy. The Halothiobacillus Neapolitanus C2 is a carboxysome that contains ribisco or ribulose biphosphate carboxylate, which helps in carbon fixation. When sulfur-rich bacteria oxidizes sulfur, it creates a carbon dioxide product. And bacteria are full of proteins enhancing this carbon dioxide fixation, and it produces energy whenever it is constructed. So if you take one bacteria and you utilize the sulfur oxidizing components making carbon dioxide, it produces a small burst of energy. So if you take millions and millions of the bacteria, then you can create a ton of energy. The United States has to go across seas to get oil from other countries. If this bacteria could be utilized in a way that it could produce their own bacteria, then it would be a tremendous discovery for not only the United States, but also the world. Annotation is the process of determining the location and functions of all the identifiable genes in a genome. So, with my project, I basically annotated the Halothiobacillus Neapolitanus C2 to discover the functional genes in the genome that I had. In my project, the materials that I used was a computer database, the PubMed search engine, an integrated microbial genome database, Google search, NCBI BLAST, NCBI CDD, T-Coffee, TMHMM, Signal P, P-Source, Phobius, PFAM, PDB, KEGG, and Metapsych. With these materials, I found, as you can see in these diagrams, I found different parts of the genes that are being used by proteins to find these identifiable genes in the genome. And in these methods, I constructed basically a poster board that summarizes most of what I did in my project. In Module 1, it consists of basic information about the gene and contains the DNA coordinates, DNA sequence, protein sequence, and also the isoelectric point. In Module 2, it gives the sequence-based similarities within the data according to the BLAST, CDD, and T-Coffee results. Module 3 gives the cellular localization data showing TMHMM, Signal P, P-Sort, and Phobius. And Module 4 focuses on the alternative opening reading frame if one is needed, showing IMG 6' sequences and finding alternative start codons. Module 5 gives the structure-based evidence revealing PFAM and PDB. Module 6 contains gene duplication, degradation, giving the IMG evidence for paralogs and sweater genes. And Module 9 consists of the proposed annotation of the gene. Out of my results, the 28 genes, all of the annotated proteins were able to be identified with 2 unnamed protein products using the modules and methods that I just said. And in my conclusion, there were 28 proteins, and all of them were identified, and the functions of them were identified. The Halothiobacillus Neapolitanus C2's functional proteins can be studied in depth, so the role of carbon fixation can be mimicked. Thank you.